title of our experiment for today is Phytoremediation in Controlling Soil Pollution. We are going through a few steps in this experiment and also we are going to see the result in the end of this video. For this experiment, our problem statement is:
Are phytoremediation plants effective in controlling soil pollution? Through this whole experiment, we aim to study the effectiveness of phytoremediation plants in controlling soil contamination. Hypothetically, we predict that the roots of water spinach can absorb nutrients such as ammonia in soil.
manipulated the presence of water spinach, responding reading of ammonia in the soil at the end of the experiment, constant mass of black soil.
5 kg soil, 2 plastic pots, food waste, water spinach, 2 mol potassium chloride, distilled water, ammonia test kit, measuring apparatus. A thin layer of 5 kg of black soil is flattened on a plastic sheet and dried under the sun. 2 kg of black soil is put into a plastic pot. Another 2.5 kg of black soil is put into another plastic pot. Both plastic pots are labeled as A and B respectively. About 7 grams of black soil from pot A is put into a container. 50 ml of 2 mol potassium chloride solution is mixed into the container. The container is shaken. The mixture is filtered into another container. The filtrate is then transferred into a bigger container. 20 ml of distilled water is poured into the filtrate. The concentration of ammonia in the filtrate is determined using an ammonia test kit. 8 drops of liquid from bottle A are dripped into the filtrate. Then 8 drops of liquid from bottle B are dripped into the filtrate. The container is shaken and left for about 5 minutes.
The filtrate is then transferred into a clearer container to better see the color change. Based on the chart, the ammonia content is 8.0 ppm.
Two kg of food waste is mixed with soil in pot A. 2 kg of food waste is mixed with soil in pot B. Water spinach are transferred into pot A while pot B is left without any water spinach as a control set. Both pots are placed in a warm but sheltered area and watered with equal amount of water every day for 2 weeks. 7 grams of black soil, 50 ml of 2 mol potassium chloride mixture is shaken and then filtered. 20 ml of distilled water is poured into the filtrate. Ammonia content is determined using ammonia test kit. 8 drops from bottle A, 8 drops from bottle B. Filtrate is shaken and left for 5 minutes. The filtrate is then transferred into a clearer container to better see the color change.
Based on the chart, the ammonia content is 0.5 ppm.
Same processes are repeated to determine the ammonia content of soil in pot B.
Based on the chart, the ammonia content is 8.0 ppm. And these are the results that we obtained from this experiment. Now let's discuss a few questions. First, why must the soil sample be dried before the experiment begins? The answer is to remove the water content and get an accurate measure of dry soil. Second, what is the significance of using water spinach in this experiment? And the answer is, water spinach can treat soil by absorbing the ammonia from the soil to ensure the ammonia content in the soil is always ideal for plant growth.
As conclusion, our hypothesis is accepted. The roots of water spinach are proven to be able to absorb nutrients such as ammonia from the soil.